Welcome, Jeremy.
Welcome.
Let's see if you could share your screen.
Can you see it? Can you see it, Bree?
Yep, perfect.
Excellent. Well, thank you for that introduction, and I really appreciate you having me today. Oh, my name is Jeremy Hartsock, and today I am going to tell you about some of the research that I'm currently working on, and also I just want to really help introduce all of you to some macrophytes that you're likely to encounter if you're out on Michigan water bodies. I'm gonna turn off my camera just so I'm not a distraction. Okay, so here's the outline. The first half of the talk is going to be what I call building an appreciation for aquatic plants. I'm going to show you a lot of common plants you're likely to see out on Michigan inland lakes. I will share with you some common aquatic invasive species that I see quite a bit. The second half of the talk, I will talk about the research that I'm doing, which is involved with developing a point intercept inland lake sampling protocol. I'll show you some of our preliminary findings, and then I will share with you our plans going forward this summer and next summer. So I always like to start things off with a picture of a neat macrophyte. This is white water lily, and behind it is bullhead pond lily. But I didn't start studying these plants until about 3 years ago. Historically, I've always been a terrestrial wetland ecologist, but goodness, these last 3 years have been amazing studying these aquatic macrophytes, and I just really gained an appreciation for them and how cool they are. So what I want to try to instill in everyone is an appreciation that these are underwater forests. They're not very accessible, so you don't see them this way, but that's what they are. They have clear structure. Just look at this scene, this underwater forest, and the key players. Just to name some, we have variable leaf water milfoil. This is Myriophyllum heterophyllum. Here we have American eelgrass, Bellisneria americana. Here's Canadian waterweed, Elodea canadensis. Here's bullhead pond lily, Nuphar variegata, and then floating leaf pondweed, Potamogeton natans. So they're underwater forests, and if they are the submergent zone, littoral zone communities, they're often underappreciated and overlooked as being part of a wetland, but they're especially important, especially if you think about Great Lakes coastal wetlands where water levels fluctuate. You know, submergent zones during low water times will develop into emergent zones, and emergent zones during high water times will turn into submergent zones. But they're very important habitats. And just look at this picture. You can see this submergent zone community, and just imagine how many critters are using this area for habitat. What do you care about aquatic plants? Well, as I said, they're doing a lot of things. They oxygenate the water by doing photosynthesis. They improve water quality and clarity. These macrophytes absorb nutrients like nitrogen and phosphorus. They provide habitat and food for fish, wildlife, and other aquatic organisms. Some macrophytes are rooted, so they are stabilizing sediment. They help to reduce wave action. And this is a big one. A diverse community of aquatic plants can lower the risk of invasion. Now I just want to introduce you to some common macrophyte species. I can't go over all of them for the sake of time, but I just want to go over some of the big important groups. And I want to make a plug. This is the main aquatic field guide I use, Aquatic Plants of the Upper Midwest. It's by Paul Skowinski. It's a great resource. I've got the link here in the bottom of this slide, and I use this one about 99% of the time. The other great one to have is through the looking glass. Make sure, though, that you get the second edition, not the first edition. So in Paul Squincy's guide, he's got about 150 plants that he talks about. And let's start with Cara. Now, in Squincy's book, he covers 8 species. This one here is probably the most common. This is, I think it's Cara contraria, but Cara is a macroalgae. It's not a vascular plant. So Cara is pretty old. On, I think, of evolutionary time. These evolved before the bryophytes. They are neat in that they can tolerate low nutrient conditions. And its common name, muskgrass, is very appropriate. And I'll tell you why. When you pull out a rake of Cara from the water, it has a distinct odor. It smells like a skunk. It's off-putting to some people. I don't mind it. But it's a really useful property to help you identify when you've got Cara. Now, a common look-alike that can confuse you. This is starry stonewort. Starry stonewort is one of the top 3 invasive species that I encounter when sampling inland lakes. Is spreading across our state. It does look like Cara. Starry stonewort is a macroalga, but it's. I'm just going to walk you through some differences. Number one, starry stonewort, it doesn't have that musk. It has no odor to it. If you look at Cara, Cara has a lot of symmetry in its branching. The branch points of starry stonewort are not even. So that's another clue. You can squish starry stonewort and it pops and it will squirt out some green goo. Cara does not do that. And the most telling indicator, if you got starry stonewort, is starry stonewort makes a bulbul. It looks like a star, and nothing else does this. So if you see that star-shaped bulbul, you know you have starry stonewort. All right, let's talk about some pondweeds, the Potamogeton species. Paul Skwinski covers 27 species. And here is an example of 6. So Potamogetons are really fun. When you kind of get a search image for the different varieties that are out there, they're not too bad to tell one apart from the other. So here, number 1, this is Potamogeton illinoisensis, Illinois pondweed. Number 2, we have Potamogeton amplifolius, large leaf pondweed. Number 3, variable pondweed, Potamogeton gramineus. Number 4, this little guy down here, this is a small leaf pondweed. I don't know the species, possibly Potamogeton pusillus or foliosus. That's kind of tough to tell. But number 5 is Potamogeton natans, floating leaf pondweed. And number 6, this beautiful one, is Potamogeton prelongus, white stem pondweed. Now, the thing with the Potamogetons, they have alternate branching patterns. So if you got something leafy, alternate branch leaves, you're probably looking at a Potamogeton. Going back to these first 3, these are they're conspicuous species, pretty common. The way to tell these 3 apart from each other, one way, is just to simply count the veins on the leaves. So in this example, here we have 8 veins on this half of the leaf. So to do the quick math, you do 8 times 2 plus 1 for the mid vein. So here we have 17 veins. And for these 3 species, when we sample, we have certain thresholds for vein counts. Potamogeton gramineus, variable pondweed, has 3 to 8 veins. When you get above that, Illinois pondweed has 9 to 19 veins. And then Potamogeton amplifolius has greater than 19 veins. Sometimes that one can have up to 40 veins. So those are some thresholds that we use to discern between these species. Let's take a look at Potamogeton prelongus, that white stem pondweed, an absolutely beautiful plant. A good way to discern this one, instead of counting veins, you can squeeze the tip of the leaf and it has this split at the apex. So this is a fun one to identify. Potamogeton prolongus, I don't know, this is subjective, but I feel it usually likes to be in slightly deeper water, like 9 to 15 feet, whereas the other ones like it a little bit shallower generally. But this is a nice population here of Potamogeton prolongus, white stem pondweed. Okay, our second AIS I want to talk about, curly leaf pondweed. So we know it's a Potamogeton because you can see the alternate branching. But the way to really know you've got curly leaf, when it's mature, it has lasagna-shaped leaves. But when it's young, they don't get this lasagna-like pattern. So what you got to look for is you got to go to the leaf margins. Potamogeton crispus has serrated leaf margins. It's the only Potamogeton that has that. So that's the indicator you look for to see if you have curly out. Another thing that's interesting about curly leaf pondweed, it has a unique phenology. Curly leaf likes cold water, so it usually completes its life cycle. It's most abundant doing its thing in June, and it can disappear by July. So if you're doing early detection studies, you want to get out on your water bodies early to find this one. And when I sample, when we're sampling later in the season, there's a lot of there are probably some water bodies that we are missing this species just because it's gone away. All right, let's talk about water lilies. I'm going to talk about two kinds: white water lily, Nymphaea odorata, and bullhead pond lily, Nuphar variegata. White water lily it has these white flowers, very beautiful. And when you see the leaves, to me, I think they look like Pac-Man. Whereas you compare that to bullhead pond lily, it's more rounded. You don't have those, pronounced Pac-Man, that Pac-Man mouth. Now, the best way to tell if you're looking at bullhead pond lily is you want to just cut the stem. And the stem, as you see in the top right corner, people say it looks like a longhorn bull. And you see those bull horns protruding out there. This one kind of looks like Yoda, but just use your imagination here. That's kind of looks like a bullhead pond lily. The flowers of these water lilies are absolutely gorgeous. This is a bullhead pond lily flower, and you can see all the pollinators in there doing their thing. Just so neat. Now, I want to just show you this. This is a white water lily, but it's a cultivated variety, and the flowers are pink. I saw this one at Shupak Lake in 2022, and this summer sampling, I found this at two other water bodies. This is a cultivated variety. It was likely released here at Shupak Lake, but if you all see this, I ask, please let me know. We're trying to track where this is at around our state. Okay, I'd be remiss if I didn't talk about water shield. This is Brezenia treberi, another floating species, and it differs from the water lilies in that it kind of looks like a football. It's got a football shape. Also, the stem, it attaches to the center of the leaf on the underside. And another common name for this one is snot bonnet. It produces a mucus on the top of the leaves that is, it's very interesting. If you get a chance, just grab one of these out of the water. You won't be disappointed. Okay, that covers the water lilies and floating species, or some of them. Let's talk about the bladderworts, carnivorous plants. And Skwinski's book, he has 8 species of bladderworts that he covers. In general, I usually only see about 5 when I'm out there. So I'll go over some. This is the most common greater bladderwort, Utricularia macrorhiza, used to be called Utricularia vulgaris. This is usually free floating. It can be down, I guess, by the substrate, but it's you just never know what you're going to get with this one. It is carnivorous, as I said. It can reproduce sexually or asexually by producing these neat turions. Always fun when you see one with a turion. A turion is a wintering bud that becomes detached and remains dormant at the bottom of the water, and then it'll germinate the following year. Next one, Utricularia intermedia. This is fun because what it does is it produces, it has a photosynthetic stem, and then it will produce a carnivorous stem that'll be buried in the substrate. So it partitions its duties to two different stems. Utricularia purpurea, purple bladderwort. This one is different in that it produces its leaves in a whorled pattern. And when I see this in the water, it has a brown color. It is just incredibly beautiful under the water, but as soon as you pull it out, it just totally crumples. It's very dainty and hard to handle out of the water. The last one I want to show you, Utricularia gibba. It's a super small bladderwort, very filamentous, not many bladders. Until you see this for the first time, you'll probably just discard it as filamentous plant material, but I see it pretty often, and it's fun to find on your sampling rake. Okay, let's cover the water milfoils, and this is a surprise. We have a lot of water milfoil species that are out there. Skowinski's book covers 10 species plus one hybrid water milfoil, which is Myriophyllum spicatum times Sibiricum. But of the 11 milfoils talked about, 8 of them are native. So there's only 3 that you've got to worry about. Just the basics on water milfoils. Milfoils have leaves in whorls, and their leaves come in whorls of 4. So at the top, we have another aquatic invasive species, the third most common one I see, Eurasian water milfoil. But look at that whorl. So there's 4 leaves here, and each leaf has what are called leaflets. And on Eurasian water milfoil, it has a lot of leaflets compared to, look at the natives, the Sibiricum over here on the top right, far fewer leaflets. And then the heterophyllum down here at the bottom right, it also has not as many leaflets. You can see the heterophyllum, the spaces between the nodes, they're reduced, they're more squished. So now, if you really want to tell the difference, if you're looking at a native milfoil versus a non-native, you got to count those leaflets. And if you can count less than 12 leaflets, that's a native species. If you have greater than 12, it's a non-native. And for me, usually the natives have 10 or less. It's pretty obvious. Eurasian milfoil, I normally count 16 to the 20 leaflets. So they really look like kind of bird feathers. I'm just going to put this up there. Take this with caution. To truly identify hybrid milfoil, you need to do genetic analysis. But in the field, if I find plants that consistently have 12 leaflets, 12 leaflets, 12 leaflets per leaf, I feel that that's an indication that hybrid milfoil is present. Also, it'll have some mixed characteristics of Eurasian and Sibiricum, but it's that 12 leaflets lets me know it's hybrid, probably. Okay, let's talk about coontails. I used to confuse coontail for Myriophyllum frequently in the water. It's really hard to tell, but when you pull it out, they do look pretty different. Ceratophyllum coontail has its leaves in whorls, but it's just it doesn't come in whorls of 4 like the milfoils. And when I see coontail, I think to myself, peace sign. It kind of has a branching pattern that looks like someone given 2 peace signs. So that's my search image indicator to let me know what I'm looking at. Lastly, we'll talk about the duckweeds. Duckweeds are native. They're a lot of fun. You'll see these in areas where usually it's maybe a little bit more eutrophic, areas with still water. Quite a few duckweed species out there. Lemna minor and Lemna turionifera and Spirodella, the ones I see the most. Spirodella, the way you tell that one is you just flip it over and it has purple on the underside and some dangling roots going down into the water. Very cute. And how about this? Wolfia, water meal. Wolfia is the world's smallest flowering plant, and it's right here in our backyard. So there you have it. That's kind of a rundown of some common native macrophytes you're likely to encounter if you're patrolling the littoral zones. I wish I could cover more, but I can say that those of you here probably know now 99% more about aquatic macrophytes than 90% of the rest of us. So I look forward, if you want, I was talking to Bree earlier that I'm happy to lead trainings to show people more species. Just I will go anywhere, anytime to talk about macrophytes. And I just want to hammer this home. These are the three most common AIS that I see doing my lake sampling. The top left, Eurasian water milfoil, leaves in whorls of 4, greater than 12 leaflets per leaf. On the bottom left, curly leaf pondweed. The leaves look like lasagna. When it's mature, you have the alternate branching. The margins are serrated. That's the key indicator. And then starry stonewort. Look for those bulbils. Nothing else does that. It doesn't smell like muskgrass. No odor with starry stonewort, and it pops when you squish it. So be on the lookout. Report those if you see them, please. Okay, we have to talk about this today. Hydrilla has been on Michigan's watch list for a long time. Last fall, it was detected for the first time in our state in Southwest Michigan. Measures have been done to eradicate this, and measures are still ongoing. It's a very problematic species. Just to show you what to look for, the leaves are serrated at the margins, and the leaves are in whorls of 4 to 8. This has a look-alike. Elodea canadensis, Canadian waterweed. This is a very common plant. I see Elodea at a lot of water bodies, and it does look like Hydrilla. They're in the same family, but Elodea, its leaves are in whorls of 3, and the margins are not serrated. Okay, so anytime you see something Elodea-ish, or you see 4 or more leaves on a node, you got to report it. Have to. Okay, so just putting this out there, there is a portion of the public that has ill feelings towards macrophytes. They can impede reclamation. I totally understand that. We've all, if you own a boat, I've done my share of having to clean macrophytes off my propeller. They can make swimming a challenge. If you have a dock, they can make getting your boat away from your dock a challenge. But I just want everyone to recognize that, these are underwater forests. They're doing a lot of good. They're great habitat. And, it's my mission as an educator to help more people appreciate macrophytes and these really neat habitats. Okay, let's transition to the second part of the talk, which is just highlighting the research that I do. The title for this is Developing an Aquatic Macrophyte Monitoring Protocol for Michigan Inland Lakes. I've been working on this project for 2 years now. Our funders are the Department of Natural Resources. These are the PIs on the project, my collaborators, Joe Lattimore, Dan Hayes, Eric Elgin, great folks to work with. But the reason why this project kind of started is there is an impetus for developing a statewide monitoring protocol that is used by as many agencies as possible. Um, Minnesota and Wisconsin are farther ahead than Michigan on this. There's kind of more of a consensus of people monitoring inland lakes the same way. In Michigan, there's a lot of folks that do lake monitoring, but we all kind of have a way that we like to do it. So we want to work to get everyone, as many people as possible, doing lake monitoring the same way to help facilitate making apples to apples comparisons. So the goals of this project, number one, establish what we call a point intercept protocol to be used by others and facilitate apples to apples comparisons. Now, a point intercept protocol for monitoring is it's perceived to be the gold standard. It's great for doing stats, great for long-term monitoring. You can go back to these same points again and again over time. So that's where it's really useful. There are shortcomings of point intercept. We can talk about that, but for doing long-term monitoring, it's really great. Other goals, we wanted to sample a bunch of lakes to obtain inland lake baseline data to better understand aquatic plant diversity, abundance, and distribution patterns in our state's lakes. We want to sample a lot of water bodies to help detect and report new locations of aquatic invasive species. And another side goal is we are working with the DNR to sample water bodies that they're also doing fish sampling at. So that way we can make correlations between the status of the plant communities and the status of the fish communities. So how does this protocol work? What do we do? I'll just run you through that quickly. For lakes that are less than 1,000 acres, we establish 100 equidistant sampling points in the littoral zone. So we choose a lake, we get the bathymetry data, we map out the littoral zone. This white here is the non-littoral deep water areas. Here is the littoral zone. We then put 100 sampling points in the littoral zone, and we go from point to point to point to point to point to point, sampling these locations. We sample every point that is in water that is less than 25 feet. At every sampling point, we throw 2 rakes from both sides of the boat. This is a unique aspect of our protocol. Some folks throw 1 rake, others throw 4, maybe north, south, east, west directions. We like 2. Our rationale is throwing 2 rakes is better than throwing 1 rake, obviously, but we have actually done the modeling to show that the benefits of throwing 2 rakes versus 1, it's a meaningful result. You're adding a significant amount of species, but you get diminishing returns as you throw that 3rd and 4th rake. You don't add as many species, and it adds a lot of time onto the survey. We want to be timely when we complete our surveys. We want them in general to be done in 1 day. If we threw more than 2 rakes, that might not be possible. We also do abundance rakings. When we throw our rakes, here is the scale we use. We have 1, 2, 3. Number 1, basically only a few plants on that rake. Number 3, that rake is completely covered in macrophyte. So to show you what a day on the water looks like, this is Long Lake in Iosco County. There is our boat in the foreground. So we do our point intercept surveys. We'll have a GPX points and a tablet to show where we got to go, and we go to all the points. And after we sampled, we can make these. This is our sampling point distribution maps. This is where we sampled in the littoral zone here. Then what we can do after we run analyses and look at the data, we can build species richness maps. So here you can see the big circles correspond to areas where there's more species that were found. At this lake, the max richness at a sampling point was 7 species. You can see the most species-rich areas are in the middle of this water body. Here are curly leaf pondweed detections. So you can see at Long Lake, curly leaf covers a large portion of this lake. This water body also has starry stonewort. This is a starry stonewort detections map. And then we had one Eurasian water milfoil detection up there in the north west corner. But yeah, this lake has 3 aquatic invasive species, the 3 that we talked about previously. We also can build abundance maps to show, where is biomass the highest, and you can see that where biomass is high, it overlaps to some degree with where the aquatic invasive species are. So this water body has it's invaded. What else do we do? This is a total species checklist for that same water body. And why is this useful? Well, look at the species that are present that are ranked based on their frequency of occurrence, the number of times that we encountered them. And you can see if you look at the top 4 species here, number 1 and 2 were Elodea, Canadensis, and Cara, native species. But of the top 4, 2 of the top 4 most frequently encountered were aquatic invasives. So that is useful for our stakeholders, that they can look at these and really come to the conclusion, yep, invasives are prevalent in this particular water body. So we make lake reports for every lake that we sample. They are publicly available. We also give the reports to DNR, to Eagle. Eagle has been very kind, and they're going to house these reports in one of their online story maps. Here's a link to that. They're not in there yet, but they should be hopefully in the next couple months. But it's great that we have a place to put these where folks can go and access these lake reports and learn about the status of aquatic macrophytes in the lakes that we sample. Also, I just want to share that at the end of the study, we are developing a database that will house all of our data. This will be publicly available. It will be R-friendly. And if you know how to use R, you'll be able to use the database to quickly summarize all kinds of information about the lakes that we survey. And we can also share that database across state lines, share it internationally. Who knows? But it is going to be publicly available. If you're interested, please feel free to email me about that. Just to give you an idea of our footprint of what we've done over the last 2 years. This is kind of a story map. So the blue dots represent lakes surveyed by Eagle aquatic nuisance control teams over the last 40 years. Eagle every year samples about 10 to 20 water bodies. That's amazing that they're able to do that, given all the other work they have to do. We were able to really get after it these last 2 years. And the red dots are what we've done. We've sampled 75 water bodies in northern Michigan over the last 2 years. And we are excited to sample more. So when you sample in the lakes, it's great. It's like a box of chocolates. You just never know what you're going to get. You may get something that is totally pristine, no shoreline disturbance, water body like this. And that's a lot of fun, but water bodies like this can also be fun too. This one obviously has more shoreline development. But yeah, you never know what you're going to get. Just to highlight the variation that we have, how different fisheries in lakes are from one another. This is a map showing species richness, or the total number of species observed at each of the 75 lakes we've sampled. So you can see that some lakes have a ton of species present. I mean, a ton is relative. Our most species-rich lakes, Black Lake and Lake St. Helens, they had 32 species present. Whereas, some, you're looking to find stuff. East Twin Lake, this is in Lewiston, Michigan. We only found 3 species. East Twin Lake has a complex history of disturbance. There was a logging mill there. It has a long-term herbicide treatment plan. So it all that has probably influenced its richness patterns and abundance patterns. But you can see the average for all of Michigan's lakes, the mean species richness is about 15.5 species. Looking at water clarity, this is neat too. Some lakes are super clear. Our most our farthest down Secchi depth was 35.5 feet at Guthrie Lake in Otsego County. Our most turbid or unclear lake was Clark Lake in Luce County. I'll show you pictures of these here in 2 seconds. But yeah, there's just a wide range of clarity in our state. So this is Guthrie Lake, blue, clear water. See down 35.5 feet, just unbelievable. But this is a surprise. This is Clark Lake. This is the one with the Secchi of 2 feet. So this is in Luce County. Clark Lake is up by Tacoma Falls. So, Tacoma Falls has root beer colored water because of DOC going into the water from surrounding wetlands. That's what's going on here at Clark Lake. So it's just DOC in the water makes it more brown. And these patches you see out here, these are actually large populations of water shield. It's a really neat site to sample. Looking at the AIS detections that we observed, 36 of the 75 water bodies we sampled over the last 2 years had AIS present. There are, some water bodies, of course, that are much more invaded than others. Many of these 36 only had like a single detection. But in general, I believe there's a clear latitudinal gradient that exists. And as you get up into the northern UP, obviously, those water bodies, they're less invaded. They're more pristine. And I hope that everyone can be responsible and we can prevent non-native species from getting into those water bodies. So that's what we've done over the last 2 years. Got a lot of data analysis to do, a lot of unique questions to ask. We're currently in the process of QA/QCing our database before we move forward with doing more analyses and before we move forward with publishing. But we'll get there soon enough. But next summer, this year's summer sampling campaign it's happening very soon. And I'm just going to share the plan, what we're going to do for next year. We are working with Eagle. Eagle has given us funding. And what we're going to do is we're going to sample 10 to 11 lakes in southern Michigan this time. And we're going to do some evaluation of macrophyte sampling approaches. So Eagle's two main approaches they use to sample lakes are the snorkel method and the ABAS survey. So all of us are going to work together to compare how the point intercept, the snorkel, and the ABAS all kind of talk to each other to see, which ones are the best for accomplishing certain goals. And maybe we can come to a consensus and have all of our teams, do the same types of survey, as I said, to facilitate apples to apples comparisons. So what we're going to do is we're actually going to do a new kind of a grid survey. We call it a point intercept heavy. So we're going to sample 300 points in every water body. And then we can model back and pretend like we sampled 200 points or see what analysis shows with pretend sampling 100 points. We can also build variable density grids. But bottom line, we want to see, is 100 points enough to do these lakes justice? So we'll find that out next summer. The Eagle teams, they will, while we're sampling the point intercept way, they'll be doing snorkel surveys. And what we want to look at here is the snorkel survey better for doing AIS detections? Are we going to, still find the same species doing both these types of surveys? And then the other Eagle team will be doing AVAS surveys. This is another type of survey that's commonly done, but we'll be comparing AVAS to point intercept again to see how those surveys compare to one another. That's going to be what we'll be doing next year. So it's really neat, that we get to work so closely with our stakeholders, Eagle and the DNR. I think it's really awesome that we're having regular conversations and we're all working together, even though we're not part of the same agencies. We all want to try to get to a point where we understand each other's methods, and hopefully we might get to a point where we might do the things, sample the same way going forward. That's really cool. So I want to give special thanks to our our funders, the DNR, Eagle, and I got to give special thanks to my team members there in the bottom right. They were just great. And I hope you enjoyed learning about macrophytes, and I am happy to answer questions.
Okay, so I am looking at, I don't see any questions that came in, but I do have questions of my own. So if you guys don't mind. So you mentioned that we should report findings of hydrilla, if we find it at lakes or on the edge. So where should we report findings of this invasive species?
Yeah, am I still sharing my screen? Maybe I have a slide. Well,
Um,
bottom line, you can report it to the MISIN website. MISIN is a great resource to report that to. You can also contact the DNR. You can contact ANC at Eagle. If you just, there's a whole lot of resources to report that stuff. And a quick Google search will give you contacts to a lot of people.
Perfect.
You can also send me an email, and then I will do the work.
Okay, great. Yep. And now we have four. So one question from Todd Losey says, how many species do you need to know to do a decent lake survey?
I'm going to say, Todd, you got to know 100, and you get there pretty quick. And you compare this to like regional supplements for wetland delineation. I mean, you got to know a lot of plants to be a good delineator. You don't got to know near as many to be a good wetland botanist. So I rounded out at 100, and then you use the book to find those tricky ones that may elude you.
So then there's another one that says, do you record only the species that are found on the rake tosses, or do you visually look into the water column at sampling locations?
Great question. We have a visual component to our survey, and we'll use that visual component when we write the species checklist for those water bodies. But when we perform the analysis, we only use plants that were detected on the rake throws. Okay, so we incorporate a visual component, but for the stats, it's only rakes. Otherwise, it just gets subjective. You know, you got to set a boundary around the boat. We're going to look at plants, 15, 20 foot away, and 15 feet for me might be different than my student. So we try not to introduce subjectivity.
Another question says, have you seen the flower of water meal under a microscope?
I have not. This is something that I want to experiment with. Perhaps you can tell I like to dabble in camera stuff with drones and underwater pictures. And my next endeavor is microscope photography. So stay tuned. This will happen.
Okay, great. There's another question that says, thank you so much for sharing this knowledge. Do you do any distribution, density measurements, or environmental data with Sonde for your sampling?
Can you say that again, Bree?
It
Sorry.
says with a Sonde, SONDE. I'm not sure what that
I think
is,
they
but
might be referring to if we use maybe sonar to
okay.
evaluate biomass. Maybe.
Yeah, that's probably, it says, do you do any distribution, density measurements, or environmental data with a SONDE for your sampling? So yeah.
I would say no, because but I know the sonde is also I think a water sampler too that takes a whole bunch of parameters. We do collect water samples to look at electroconductivity, the pH, and alkalinity. We are interested in this whole sonar abundance technology. I don't know if we're going to be able to implement it. It does cost a lot of money, but we do recognize its value. But we don't do it yet. We just use our 1 to 3 rankings. It's easier, it's quick. It still tells a decent story.
So another question came in. It says, how long did it take you to learn these species?
I'm still learning. In fact, I have a training scheduled with Paul Skowinski to help our whole team get better identifying charophytes. So as you know, it's a lifelong journey learning these plants. But for me to really get comfortable, um, uh, it took a whole summer, and you really need to see a lot of plants at different points in the year as well, because they change a lot early season to late season. Um, Variable pondweed, its common name is very appropriate. That plant is highly variable. It can be very small, it can be big. So, but a full year of practice got me good. Um, Still got a lot of work to do.
Excellent. So there's a couple more. It says, does your research focus primarily on species richness, or you do water sample as well for DOC nutrients?
We don't do sampling for DOC, and yeah, we do the most cursory assessment of nutrients, by just looking at conductivity. That's not very good. Um, it just comes down to cost constraints. Those analyses cost more money, and we just have to sample as many lakes as possible, as cheaply as possible. And the other part of the question was the analyses that we're looking at, correct? So we are concerned about species richness comparisons across the board. I'm a plant community ecologist, so eventually I will start exploring how the communities differ across all the inland lakes. And, are there effects of long-term herbicide treatment? Are there effects of long-term development? So we'll be able to tease out all of these intricacies that are potentially affecting the communities um, of these water bodies. That's the long-term plan for the next year and a half.
Excellent. It says, besides the three invasive species you discussed, did you encounter any other invasive plants?
Um, of course, you see Phragmites, invasive typha, purple loosestrife pretty regularly. We don't record that on the data sheets. Other ones that we encounter, there's some, I don't want to call them invasive, but there are some non-native naiad species. Um, and I didn't cover the naiads, the Nias, Najas, the Nia species. There are some non-natives that we encountered. Um, the the pink water lilies, like I said, please report those if you see them. They're not listed as an AIS, but we want to know kind of where they're at in case they do become problematic. But no frogbit this year. It is present at Fletcher Pond, a water body we sampled, but we did not see it. So that's good.
It says, did you ever run into Vasey's pondweed?
Faces. Oh, did not. Um, I have not seen that one. The only time I've heard about it is when I worked for the Great Lakes Coastal Wetland Monitoring Program. My buddy found it in the UP, but I know about it, would love to see it, haven't yet. But I know that's a threatened one, so my eyes are always open.
Okay. And then it says, have you done any studies to assess the efficacy of the rakes with different species of vegetation?
Yeah, so that's the cool thing about the two rake throw approach is we can calculate detection probability. And some species have a higher detection probability than others. So the big leaf Potamogeton amplifolius, for example, if it's there, usually it's caught on both rakes. In contrast, like Potamogeton gramineus, its detection probability is something like 33%. So with that two rake throw, we can model detection probabilities for all the species, and that is going to be a component of our future publications, looking at which ones you're more likely to detect, which ones not so much.
Okay, great. And then let's see, there's a few more. It said, do you use environmental DNA technology? I think you may have answered that, but
Not this year, but in 2025, our next project after this summer, the following summer, we're going to be sampling lakes for uh, the Huron-Manistee Forest Service. And when we encounter hybrid milfoil, we are going to send that to what I believe is the University of Minnesota for genetic evaluations, which is really important because you want to find that variant that might be resistant to herbicide. So we'll be doing that in 2025.
Okay. Let's see. Here's a specific question. It says the property I manage has a 3-mile shallow, less than 10 feet, river connecting 2 lakes. Do you have any recommendations for sampling the rivers versus the lakes they connect? Example would include snorkeling, is that best, etc.
Um, I would say, depending on how deep that connection is, um, just put a rope on a double-headed rake and toss it in there at said 3 miles. So depending on how much work you want to do a reasonable spacing and see what you get. That's how I would approach it.
Okay. Let's see. It says, do you incorporate any wetland vegetation or only aquatic? And I think you
Um,
answered that too.
only aquatic. Um, otherwise, it's just the job becomes too big.
Okay. Um,
I will say that we do list Sagittarius species. Persicaria amphibia, water smartweed, has kind of an emergent growth form, and it looks different when it grows out in the water. So, we log those species when they are in open water.
and then another one says, do you see certain aquatic species more often or exclusively in rivers?
Yeah, one Potamogeton example that you see a lot more in rivers is Potamogeton nodosus. I don't know what the common name is off the top of my head, but it likes flowing water. So I see it at my house on the Grand. It's a real treat. I just know where there's a nice population and it's just likes moving water.
Great. Excellent. That's interesting. And then it says, are there any specific species you'd recommend for living shoreline installations?
Like for shoreline restoration, is that the context that it's coming from? Say lining shorelines?
It says living shoreline installations. Sorry, I read it
Living
wrong.
shoreline installations.
So I guess
I
it's
think,
probably saying like, what species would you propose for?
yeah, I think the best approach would just be to do an assessment of look at the natural shorelines on your water body. And every lake is different. So pick species that are present at your particular site and go from there. So that's a good best management approach. Shop local.
Okay. Let's see. Any more questions? I think that might be it. Um, thank you so very much. This was a very informative webinar, and I liked all your, cheat sheet of, how to ID some of those tricky plants. So we do appreciate it.
Well, thank you for having me. I really appreciate uh, all you guys do.
Thank you so very much. And we hope to have you do get out in the field and do a training again. So thank you
That
so much
sounds
for your time.
great. And please don't hesitate to send me an email if you ever have a question about plant identification or doing trainings. The door is always open.
Perfect. All right, Jeremy. We'll see you later. Thank you so
Thank you very
much.
much. Take care.